Hello. Very good evening to the student who have joined. Can you let me know in the chat box if my voice is audible clearly, and also if you can see my presentation and my video. Just let me know in the chat box if everything is alright with the presentation, video and audio. Okay, thank you so much. We will wait till more students join, then we will start today's session. Okay, so I think I can start with the introductions, and I hope my voice is audible. If it is not, please let me know. Okay, so I think I will wait for 1 more minute, and then I will start the today's session. Meanwhile, if there is any problem while listening to my voice or seeing the PPT, please kindly let me know. At 6 p.m. I will wait for 1 or 2 more minutes, then I will start the session. Okay, so very good evening to the students who have joined. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. Today it is the week 11 live session. So again I will go with the introductions. So very good evening to the students. I hope my voice is audible and you can see my video and PPT clearly. If not, please let me know in the chat box. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. Today it is the week 11 live session for the NPTEL course of Organic Chemistry in Biology and Drug Development. So before starting the session, I would like to say a few things before starting the session, like always. So I hope the PPT is visible to you. I have made it full screen, and if you cannot see the PPT, please let me know as soon as possible. Okay. So today it is the week 11 live session for our course Organic Chemistry in Biology and Drug Development. Each week we are going ahead with a week. So we started our first week live session. It was a combination of week 1 and week 2 course content. So today it will be the week 12 course content. Today's session I will discuss the week 12 course content. Right now the last week means the week 12 course content is online. So today we will discuss the questions and the topics which are related with the week 12 course content. These questions will help you to understand the course better, and also it will help to for your assignment purpose. Now your final examinations are also closed, so it will be better if you pay attention to your previous classes also. Before starting, I would like to say few things. First, there will be a problem solving session every week, and the sessions will be on every Tuesday from 6 to 8 p.m. These sessions will be live on Google Meet platform. If any network disruption happens, please wait for a few minutes. Do not leave the session. I will join as soon as possible. So if any network issue happens during or from my side, please bear with me, wait for a few minutes, do not leave the class. And if the network disruption happens at your side, you can rejoin again, and if it does not solve, you can I will share the lecture video and also the PPT for your convenience. So don't be afraid that you will lose your connection. And during the session the students can post their question, doubts and everything through the chat box. So the chat box is hello. Yeah, is my voice audible? I think there was some network issue. I will present it again. Okay, so is my voice audible to you? Yeah, sorry. So I have said that there may be any network disruption. Plus, do not leave the class if there is any network problem. I will rejoin as soon as possible. Okay, and because there are some network issues here today, so bear with me and please do not leave the class. Okay. So during the session, the students can post their question, doubts, everything through the chat box. We are, yeah, there was some network issue. Right now it is fine, right? You can hear my voice, or network issue is still there. And also, can you see the PPT? Yeah. Okay, thank you so much. Yeah, I am looking at the chat box every time. So if there is any problem, any doubt, if you cannot listen to my voice and the PPT, you cannot see the PPT, just drop a message to the chat box. Okay. And if there is any doubt or query after the class, the students can drop an email to me and I will answer their query through email also. And as I have said, this presentation and recording will be shared to the students soon. So as I said, today it is our week 11 live session. So we are going ahead with each and every week. Okay, PPT is right now, it is visible. Oh yeah, I hadn't shared it before. So I think now everything is fine, right? You can see the PPT and also you can listen to my voice and you can see the video. So yeah, I think everything is fine right now, and if you cannot see the PPT, please let me know. Okay. So today it is our week 11 live session, and here we will discuss week 12 course content. So what is in your week 12 course content? We have cancer and chemotherapy, we have anti-cancer drugs, aromatase inhibition and anti-ulcer drugs, cholesterol lowering agent, cholesterol biosynthesis, pharmacokinetics and pharmacodynamics, and QSAR principles. So actually this course content which is in your week 12, it is very important in the drug development purpose. Okay. So as every time I will show you the question, you have to try to answer that question and tell me which of the following options are correct among 4 options. Okay, so before starting the session, I want to tell you one more thing. So today it is a week 12 course, we will discuss the week 12 course content. So it is the last course, last week's course content, right? So after that we will have 2 more classes actually. One class will be on next Tuesday, on 17th of October, which is our usual class. That will be the last Tuesday class which we have. It will be from next Tuesday 17th October from 6 to 8 p.m. and one more extra class I will be needing. So what I will do is, I will summarize your whole course. Okay, there will not be much MCQs. I will not discuss much MCQs there. I will have a few MCQs to see if you are if you remember what I have told you or what we have read till now. To see that, I will give you few MCQs in next Tuesday's class. But before that, next Tuesday's class, what I will do is, I will take 1 hour extra class. That extra class I have scheduled is on your 15th October. So I am written it here. Okay, and you will be getting the email notification. So it will be on 15th October 2023 on Sunday. Okay, and it will, the timing will be 11 to 12 in morning. Okay, so it will be on from 11 a.m. to 12 p.m. this Sunday. Okay, 15th October. So in this 1 hour class, I will try to summarize few things about your course. I will try to make you remember the things we have done in the previous classes. Okay, like for protein synthesis, like from enzyme inhibition and all those things, the DNA, RNA replication, translation, these things. And on the next Tuesday's class, which will be our last class for this session, for this July session, that will be on 17th October. So in that class, I will summarize the things left for our from the Sunday's class, and then I will show you few questions, and I will expect you to answer the questions clearly, because this will be the last session before your examination, and you have to revise your things, right? So just I have to tell you these things. I hope you will remember, and I request you, please join this summary classes. Okay, it will be beneficial to you, since we are doing these classes from previous 12 weeks. Many things you may forget, so I will try to make you remember the things, so it will be helpful for you. Okay. So now I will start today's session. So the first question of today's session is problem number 1. It says that palbociclib is an inhibitor of dash kinase. Your options are number A, JAK, number B, BCR-ABL, number C, CDK4 or 6, and number D, MET. So kinase, let me tell you, kinase is a particular enzyme which actually catalyzes the transfer of the gamma phosphate of a gamma phosphate from ATP to a particular protein or particular side chain of protein. Okay, so this is the role of the kinase enzyme. Now kinase inhibitors means the molecules which inhibit the function of the kinase are a very famous class of anti-cancer drugs right now. They are very important. Okay, they are getting very much importance because they are getting approved by FDA recently. Many kinase inhibitors are getting approved for the as anti-cancer drugs, mostly anti-cancer drugs. So why these kinases are act as an anti-cancer drug, these things I will discuss on the next class, on the summary class. Okay, I will give you a detail about it. So this question is related to the kinase inhibitor, and kinase inhibitors, this palbociclib is also a very important kinase inhibitor for cancer as an anti-cancer drug. So you have to identify this inhibitor is of which class of kinase, which family of kinase does this palbociclib inhibits. Your options are number A, JAK, number B, BCR-ABL, number C, CDK4 or 6, and number D, MET. Try to answer the question in the chat box. A, B, C or D, which of the following you think is the correct one? If you don't know the answer, then it's also fine because there is no marks in it. But if you know, or if you want to try, you can try to answer this question. Let me check the chat box. Okay, so I have two answers which is saying it is C. Okay, let me check and let us discuss. So the correct answer will be, very good, it is CDK4 and 6. So what is a CDK4 and 6 and how does it work? Okay, so basically this is a cell cycle. This is a cycle through which a particular cell undergoes the cell division. Okay, so the first step is the G1 step. Here the cell accumulates the resources needed to duplicate the genome. After that, after the G1 state, the particular cell can go to a resting phase, okay, which is the G0 phase. It is a quiescent state, which is a resting state, and sometimes the cell do not go to the resting state, it directly go to the S phase, which is the actual duplication of the genome occurs, the DNA synthesis occurs in the S phase. And then what happens, like it goes to the, and there is a G2 state. Okay, so what happens during the G2 state, proper DNA replication is checked and final preparation of mitosis phase, and then it goes to the mitosis phase. What happens during the mitosis phase is the chromosomes are separated and the actual cell division occur. So now what does this CDK4/6 inhibitors do? They actually inhibits this particular phase. Okay, this particular cycle, it is inhibited by this CDK4/6 inhibitors. It may be CDK4 or CDK6 enzyme which are actually necessary for this G1 phase. Okay, and these inhibitors they actually inhibit this particular state, so cell division do not happen. So how this kinase inhibitors, they work as an anti-cancer drug. So before starting, we will go to this Pablocycillib. So this is the structure of Pablocycillib. So as you can see, in this bluish, we have this pharmacophore for this particular compound. Okay, so the primary target is CDK4/6. It is a type 1 kinase inhibitor. So there are several kinds of kinase inhibitor. We have type 1, we have type 2 A, we have type 2B. So all these things I will discuss later in the summary class. Okay, so and it binds to the ATP pocket in an active configuration through 2 hydrogen bonds with a kinase hinge valine 101, one with aspartate 163 of the DFG motet. Its cyclopentyl group occupies the ribose pocket. Okay, so the treatment of hormone receptors of the use of this particular drug is for the treatment of hormone receptor HR positive, HER2 negative advanced or metastatic breast cancer in postmenopausal women, and the side effects are neutropenia, leukemia. So how the kinase inhibitor works? So basically in the kinase, the particular enzyme, there is a particular phase which is called the hinge region. So the kinase inhibitor actually binds with that hinge region of this enzyme. And why it binds to the hinge region? Because it is the ATP binding pocket. So to inhibit a particular kinase enzyme, we have to design a molecule in such a way it occupies the position where the ATP binds with the active enzyme, so that ATP cannot bind and the kinase enzyme cannot work. And why kinase inhibitors are a very good class of anti-cancer compound? Because kinases are, recent studies have shown that kinase enzymes are very much involved in various cycles or various cell signal regulations of the your malignant cells. That is why kinases are becoming a very good target for the anti-cancer drug design. So this was the first question. Now I will go to the next question. So problem number 2 says, identify the incorrect statement about mitomycin C. Your options are: number A, it contains a azacitidine ring; option B, it is a prodrug; option C, the first step in its method of action is reduction by NADPH; and number D, it forms an indole-hydroquinone intermediate. Mitomycin C is also a very important drug. So you have to identify which of the following statement is incorrect regarding this particular drug mitomycin C. Number A, it contains a azacitidine ring; number B, it is a prodrug; number C, the first step in its method of action is reduction by NADPH; and number D, it forms an indole-hydroquinone intermediate. Which of the following statement you think is incorrect? Let me know in the chat box. Identify the incorrect statement about mitomycin C. Your options are: number A, it contains an azacitidine ring; number B, it is a prodrug; number C, the first step in its method of action is reduction by NADPH; and number D, it forms an indole-hydroquinone intermediate. Let me check the chat box. So I have A or B. So I have two answers. Now let me see which of the following is the correct, A or B. So the correct answer means the incorrect statement regarding mitomycin C will be number A. It contains an azacitidine ring. Now let us see what is the structure of this mitomycin C. So here it is the structure of the mitomycin C. So as you can see, the structure is little bit complicated. We have so many chiral centers here, and that's why this molecules designing synthesis are very tough, because we have so many chiral centers here. So let us see first what is the, so this is the mitomycin C. So as you can see, here, this is the first, this mitomycin, it converted to mitomycin hydroquinone. Okay, so this is the prodrug. So number B statement is correct, because this drug is not an active drug. This is basically a prodrug. This quinone, this is the quinone part. So it is a part of a prodrug. Why? Because it is more stable. So first, this prodrug has to be activated to the active form, which is the mitomycin hydroquinone. And the enzyme which takes part in this transformation is NQ01 reductase or NADPH or CYP450 reductase. Okay, and what does this particular reductase do? So this particular reductase also converts this H2O2 to hydroxyl radical. So there is a Fenton reaction happens here. Okay, and this generates the cytotoxic hydroxy radicals during this conversion. Okay, so now this mitomycin hydroquinone is converted to indole-hydroquinone intermediate. This is the indole-hydroquinone intermediate. So as you can see here, this group is unsaturated. So after we have this this is a saturated center, 5-member nitrogen ring. So after this unsaturation, it is converted to a indole group, which is the indole-quinone, okay, intermediate. So so the first step is the method of action is the reduction by NADPH. It is also correct, and it forms an indole-quinone intermediate. It is also correct. So now all this conversion. So after that, this indole-hydroquinone intermediate, it forms a mitomycin DNA adduct, which is the cross-linked DNA, and that's how it actually binds with the DNA, and this acts as a anti-cancer drug. So why the first statement is incorrect? It says it contains a acetidine ring. So as you can see, there is a particular ring, nitrogen containing ring, which is 3-member ring here. So what is this name of this particular 3-member nitrogen containing ring? The name is your acetidine. Acetidine means the 4-member nitrogen containing ring, and if the ring is 3-member nitrogen containing, they call acetidine. So that's why the first statement is incorrect, because it does not have a acetidine ring, it has a acetidine ring. Okay, so as you are studying the organic chemistry, the interface of organic chemistry and biology, so it is very important for you to remember the names of the heterocyclics, especially the nitrogen containing heterocyclics. In the biological system, in the drug development or the biology chemistry, nitrogen containing heterocyclics are the most important. You will see in our bodies also, we have nitrogenous bases in the DNA. The proteins, they contain the nitrogen, the amino acid side chains always. So nitrogens are a very important part. Nitrogen containing heterocyclics are most the drugs. I think almost 80 to 90% of drugs, they contain nitrogen containing heterocyclics. And in fact, this nitrogen containing heterocyclics are a very important class of various drugs. It may be anti-cancer, it may be antiviral, okay, it may be the antifungal. All these drugs, they contain the nitrogen containing heterocyclics. So remembering the names of nitrogen containing heterocyclics are very important. It may be the 3-member, it may be 4-member ring, 5-member ring, saturated, unsaturated, all the things you have to remember. Okay, it will be only better for you if you remember the names of the particular nitrogen containing heterocycles. Okay, so this was your problem number 2. So now I will move toward the next problem. So our problem number 3 says, which reaction is involved in the method of action of calciamycin? Okay, your options are number A, Baylis-Hillman reaction, number B, Chan-Lamb coupling reaction, number C, Diels-Alder reaction, and number D, Birchman cyclization. Which reaction is involved in the method of action of calciamycin? Identify which of the following you think is the correct one. Number A, Baylis-Hillman reaction, number B, Chan-Lamb coupling reaction, number C, Diels-Alder reaction, and number D, Birchman cyclization. All these are very important name reaction of organic chemistry. While you are reading the organic chemistry in biology and drug development, it is very important for you also. So to remember the organic name reactions, because name reaction constitute a very huge part of organic chemistry, which is a very important one actually. So tell me which of the following you think is the correct one in the chat box. Which reaction is involved in the method of action of calcimicine? Let me check the chat box now. Okay, so I have few answers and all of the answers are saying it is number D. Okay, let us check. So the correct answer will be, very good, it is number D, Bergman cyclization. So let us discuss first what is Bergman cyclization. So basically it is a reaction, it is a radical cyclization. Okay, so it goes to the birth of the radical or photochemical conditions of it can be on heat or H nu. Okay, so there is a indiene. So this is called the indiene. Why it is called the indiene? In means ene, in means your double bond. Okay, so this word, this terms you have to remember, ene, this means your double bond, and ine, y ne, it means your triple bond. So as you can see, there is one double bond and two triple bonds, right? So it is called the indiene. So wherever there is this kind of indiene, what can happen? There can be this kind of cyclization which gives us a benzene ring basically with two diradicals in 1 and 4 position, which is called the 1,4-benzene diol or 1,4-benzenoid or parabenzene diradical. Okay, and this diradical can also form this kind of isomerized indiene, or it can, what it can do is it can take up hydrogens. Okay, and after taking hydrogen, what it can do is it can form this kind of it can take a hydrogen from this group, okay, which have a two this is cyclized cyclohexane which has a two double bond, okay, and it will be forming benzene, and this kind of group we will have. So it is known as cycloaromatization. Okay, so this is known as cycloaromatization. So now what is the mechanism of action of calcicimine? So this is the structure of calcicimine. As you can see, we have this kind of indiene functionality here, we have a sugar moiety here, and we have this kind of disulfide bonds, okay, present. So first what we will have, so first the nucleophile, there will be an external nucleophile which will attack here, and we will have open thiol group, and now this thiol's nucleophile or lone pair, it will attack on this okay, hello, is my voice audible to you? Just I am checking it between. Can you listen to my voice properly? Please, can anybody can let me know in the chat box. There may be some internet issues, so I am just confirming if it is fine. Okay, yeah, so there is a little bit of internet disruption here, so bear with me. So yeah, where was I? I was here, like this calcisamine, it has a indine functionality. So first, this is a product kind of structure which is activated by a nucleophile, and we have this free thiol group, and this lone pair of thiol, it will attack on this double bond, because we have a unsaturated alpha beta unsaturated ketone here also, as you can see. So it will be attacking on the beta carbon, okay, and we will have this kind of tautomerization here. We will have this kind of tautomerization here, okay, and after that we will have a Bergman cyclization. So after this part we will have a Bergman cyclization, which will be forming a diradical. Now this diradical will be acting on the DNA, and it will form the DNA diradical, which will give rise to DNA double strand cleavage. So that's how it cleaves the DNA, and it shows its anti-cancer activity, okay, by cleaving the DNA for the malignant cells. And so one thing you can ask, like, what is the reason why the particular this kind of drugs acts on the DNAs of only malignant cell, not of the normal cell. So this kind of question arise many times, and this is a very important question while designing a particular anti-cancer kind of drug, because cancer means it will be a set of malignant cells, okay. So this kind of drugs, it can also cleave the DNAs of the normal healthy cells. So there may be a selectivity issues there, so we have to be thoughtful while designing anti-cancer drugs, okay. And the reason why it does not works in that way is, like, there is some physiological differences between a particular cancer cell and the healthy cell, okay. There is a pH difference, there are very much some cell signaling differences. So while designing this kind of drugs, we have to be thoughtful about those differences, so that the drug only work on the DNA or any on the only on any machinery of the malignant cell, not the normal healthy cells, okay. Okay, so this question was related to the calcimicine. Yeah, so now I will move toward the next problem. So the problem number 4 says, which of the following is not involved in the treatment of PUD, okay, and PUD, the full form is of PUD is the peptic ulcer disease. Your options are: number A, amoxicillin; number B, famotidine; number C, misoprostol; and number D, levocetirizine. So which of the following drugs is not involved in the treatment of PUD? This is a kind of easier question. If you read the options carefully, I think you can answer it within a minute. So try to answer in the chat box A, B, C or D, which of the following you think is the correct option. So I will repeat the problem number 4 again, which says, which of the following is not involved in the treatment of PUD. Your options are: number A, amoxicillin; number B, famotidine; number C, misoprostol; and number D, levocetirizine. Which of the following is not involved in the treatment of PUD? Tell me in the chat box which of the following you think is the correct one. I will discuss it in detail after I show you the answer. I have few answers. I have number A, number D. Okay, let me show you the answer and then let us discuss. Okay, some new students also joined, so I will give you one more minute. Which of the following is not involved in the treatment of PUD? PUD means peptic ulcer disease. The options are number A, amoxicillin, number B, famotidine, number C, misoprostol, and number D, levocetirizine. Let me check the chat box again. Okay, let me show you the answer and then let us discuss. So, the correct answer of this question will be number D, levocetirizine. So, levocetirizine is not a drug which is involved in the treatment of PUD. Levocetirizine is basically an anti-allergic drug. Okay, it is basically given when you have an allergic reaction. Okay, it works on that particular allergic receptors of our body. So, levocetirizine is not involved in the treatment of peptic ulcer disease. So, what is the drugs that are used in the peptic ulcer? So, we have a number of classification for the drugs which are used for the peptic ulcer. The first one is the gastric acid secretion inhibitor. So, some drugs can be used as gastric acid secretion inhibitor, which will inhibit the secretion of excess gastric acid in the stomach. So, there are 4 types of these particular gastric acid secrete inhibitors. First one is the H2 antihistamines. Okay, and the option examples are cimetidine, ranitidine, famoxetidine, and roxetidine. There are anticholinergics, and this actually works on the cholinergic receptors. Okay, and the options are pyrazipine, propanthalin, and oxyphenolime. There are proton pump inhibitors which works on the proton pump of the stomach, and the options are the examples are omeprazole, esomeprazole, pantoprazole, lansoprazole, rabeprazole, and dextromethorphan. Okay, and there are prostaglandin analog like misoprostol. So, these drugs and there is a gastric acid neutralizers which are antacids, which are very famous treatment of the we all know we are taking antacids now and then whenever we have this acidity problem. So, they will neutralize this particular excess gastric acid which is secreting in the stomach. The options, the examples are systemic and non-systemic. The systemic ones like sodium bicarbonate and sodium citrate, and non-systemics like magnesium hydroxide, magnesium trisilicate, aluminium hydroxide, magaldrate, and calcium carbonate. We have ulcer protective, and these are sucralfate and colloidal bismuth subcitrate. And anti-H. pylori drugs, this anti-H. pylori is a particular bacteria which is associated with the peptic ulcer, and this anti-H. pylori drugs consist of several antibiotics like amoxicillin, clarithromycin, metronidazole, tinidazole, tetracycline, CBS, these kinds of drugs. Okay, CBS means colloidal bismuth subcitrate. Okay, so these are the classification of particular drugs for peptic ulcer. I will suggest you to remember the classes at first, like there are 3 major, so there are 4 major classes, like there are gastric acid neutralizer, gastric acid secretion inhibitor, gastric acid neutralizer or antacid, and ulcer protective, and anti-H. pylori. And there are various the antihistamines, anticholinergics, proton pump inhibitor, prostaglandin analogs. These are for the gastric acid secretion inhibitor. Systemic, non-systemic are the antacids, and I will suggest you to remember 1 or 2 example of each and every class. Okay, so the question can be given like ranitidine is a particular class of which kind of anti-ulcer drug, and you can be given 4 options like antihistamine, anticholinergic, prostaglandin analog, or proton pump inhibitor. So, if the question is given like this, you should able to answer that particular question. Okay. So, this was the problem number 4. Now, I will move toward the problem number 5. So, problem number 5 says, what is the correct order of intermediates in the route of the cholesterol biosynthesis? Your options are number A, HMG-CoA to mevalonic acid to squalene, number B, squalene to HMG-CoA to 7-dehydrocholesterol, Number C, mevalonic acid, farnesyl diphosphate and squalene, and number D, acetyl CoA, HMG CoA to squalene. Somebody wants to join, just let me admit. Yeah. So, I will repeat the question number 5. Again wants to join. Hello, is my voice audible to you? Something is going, there is some particular. Yes, okay, so there is some problem with this Google Meet, I think, today. So, yeah. Okay, so problem number 5 says, what is the correct order of the intermediates in the route of cholesterol biosynthesis? The options are number A, HMG CoA to mevalonic acid to squalene, number B, squalene to HMG CoA to 7-dehydrocholesterol, number C, mevalonic acid to farnesyl diphosphate to squalene, number D, acetyl CoA to HMG CoA to squalene. So, which of the following is the correct route of intermediates in the route of cholesterol biosynthesis? Let me know in the chat box A, B, C or D, which of the following you think is the correct one. What is the correct order of intermediates in the route of cholesterol biosynthesis? Your options are number A, HMG CoA, then mevalonic acid to squalene, number B, squalene to HMG CoA to 7-dehydrocholesterol, number C, mevalonic acid to farnesyl diphosphate to squalene, and number D, acetyl CoA to HMG CoA to squalene. Let me know in the chat box A, B, C or D, which of the following is the correct order. Let me check the chat box if there is any answer. Okay, so I have few answers. It is saying number B, number D, number D. Others also try to answer the question. I will give you 1 more minute, then I will show you the answer. What is the correct order of intermediates in the route of cholesterol biosynthesis? Number A, HMG CoA to mevalonic acid to squalene. Number B, squalene to HMG CoA to dehydrocholesterol. Number C, mevalonic acid, farnesyl diphosphate or squalene. Number D, acetyl CoA, HMG CoA and then squalene. So, what were the answers in the chat box? Number B and number D. So, I have number B and number D as answers. So let us check which one is the correct one. So the correct answer will be number C, mevalonic acid to farnesyl diphosphate to squalene. Now let us see the what is the biosynthetic route for the biosynthesis of cholesterol. So the starting point of cholesterol biosynthesis is acetyl CoA. The first acetyl CoA is the starting point. Okay, acetyl CoA is converted to particular. So this is very important to remember what is the starting point of cholesterol biosynthesis. The question can be given like, write what is the starting point of the cholesterol biosynthesis. So it will be acetyl CoA. Acetyl CoA is converted to acetoacetyl CoA. Okay, and what is the enzyme which is involved in this transformation? It is acetoacetyl CoA thiolase. Then another molecule of acetyl CoA joins, and we have 3-hydroxy 3-methyl glutaryl CoA, which is called the this 3-hydroxy 3-methyl glutaryl CoA is known as the HMG CoA. Okay, HMG CoA is abbreviated from of this 3-hydroxy 3-methyl glutaryl CoA, and the enzyme which is involved is the HMG CoA synthase. After that, this HMG CoA, it is converted to the mevalonic acid, and the enzyme which is involved here is the HMG CoA reductase. Now there are several biosynthetic paths. After that, this mevalonic acid goes to the farnesyl diphosphate. Okay, and after it goes to farnesyl diphosphate, the farnesyl diphosphate is converted to the squalene, and the enzyme which is involved in this particular conversion is a squalene synthase. Okay, somebody wants to join. So this, then we have this squalene is converted to the 7-dehydroxycholesterol. Okay, and after the transformation of 7-dehydroxycholesterol, it is converted to cholesterol by this 7-dehydroxycholesterol reductase. Okay, so correct order of intermediate will be this option number C is correct, mevalonic acid to farnesyl diphosphate to squalene. Okay, so for this particular question, for this particular question, I would suggest you to memorize this all biosynthetic pathway, or which is comes after which intermediate, and also try to remember the names of the enzyme. The question can be given like this, like the like acetoacetyl CoA to HMG CoA conversion is done by which enzyme. Okay, so this kind of questions can be given also from this cholesterol biosynthesis. The starting point of cholesterol biosynthesis, which is acetyl CoA, you have to remember, and which is coming after what, you have to memorize all these things. Okay, so it was the problem number 5. So now I will go to the next problem. Problem number 6 says selegiline is a specific inhibitor of, your options are number A, myo B, number B, myo A, number C, ACHE, which is your acetylcholine, and number D, ACE, which is your angiotensin converting enzyme, and myo A, myo B, these are specific enzymes. Okay, so selegiline is a specific inhibitor of number A, myo B, number B, myo A, number C, acetylcholine, and number D, ACE. Try to answer it in the chat box. All of you try to write which of the following you think is the correct option. Even if your answer is wrong, it does not matter, because there is no marks on this kind of questions, but all of you must try to answer these questions. It will be only better for you. Selegiline is a specific inhibitor of which enzyme? Let me check the chat box. Okay, so I have few answers. It is say number C, number A, number A, number A. So I have one number C and others number A. Okay, let me check what is the correct answer, but before that I will give you one more reminder, since I have said it during the first of today's session. So we will be having one extra class, okay, extra summary class, which will be on extra summary, we will having a extra summary class, okay, on 15th October Sunday, from it will be from 11 am, okay. So we will have 1 hour of from 11 am to 12 pm we will have a extra summary class where I will summarize your whole course content. So I request all of you to join that summary class. The link will be same as today's Google Meet, and you will be getting a reminder of the class before of the class. So I will request all of you, please join that summary class, okay. So now I will show you what is the correct answer for the problem number 6. So the correct answer will be, very good, number A, MyoB. So the full form of myo is the monoamine oxidase, okay. So monoamine oxidase inhibitors are a particular class of powerful antidepressant drugs, okay. So monoamine enzymes, basically there are two isotypes of, first I will just cover this part. So monoamine oxidase inhibitors are inhibitors of a particular class of enzyme which is a monoamine oxidase or myo. So these have two kind of isozyme, okay, isotypes. One is myo A and another is myo B. So myo A is the present in the cytoplasm of neuron of the like the CNS and peripheral tissues, for example liver. It acts on non-specifically on Na, 5-HT and dopamine, and chlorgyline is a specific inhibitor of myo A, okay. So myo B present mainly in the CNS, acts more on dopamine. Selegiline is a specific inhibitor of myo B, and your paraglide is a non-selective myo inhibitor. So remember few examples of first remember few examples of particular enzyme. So this chlorgyline is a specific inhibitor of myo A, selegiline is a specific inhibitor of myo B, and paraglide is a non-specific myo inhibitor, okay. So monoamine oxidase inhibitors, these are a particular class of powerful antidepressant drugs. They are particularly effective in the treating atypical depression, panic disorder and social phobia. Due to potentially lethal dietary and drug interaction, myoids have been reserved as a last line of antidepressants, okay, used only when other class of antidepressant drugs like SSRI and TCS have failed. We will discuss antidepressant class of drugs again in your summary classes, okay. I will discuss it on next Tuesday, I think. So next Tuesday also we will have a summary class. So there I will discuss this particular class of antidepressant drugs. It was not in much detail in your course, but I want all of you to understand also that particular class of antidepressant drug. So antidepressant drugs, there are several classes. SSRI, which are the serotonin receptor inhibitors, this selective serotonin receptor inhibitor, TCS, and myo A or myo I's are also a very powerful class of antidepressant, but they have a very much drug interaction and lethal dietary and drug interactions. So they are reserved as a last class of antidepressant. First a patient will be given other class of antidepressants. If those do not work, then only this particular drugs are prescribed, okay. So this was about your monoamine oxidase inhibitors. So now I will move toward the next problem. Problem number 7, it says that which of the following is not a QSAR parameter. QSAR means your quantitative structure activity relationship, okay. So which of the following is not a QSAR parameter? Your options are number A, hydrophobicity, number B, hydrophilicity, number C, steric, and number D, electronic. So which of the following is not a QSAR parameter? Let me know in the chat box whatever you think is, which of the following you think is the correct option or the correct answer. somebody wants to join. Okay, so I am have started getting the answers again. Okay, I will repeat that particular problem number 7 again. So problem number 7 says, which of the following is not a QSAR parameter? Your options are number A, hydrophobicity, number B, hydrophilicity, number C, steric, and number D, electronic. So let me check the chat box now. So number D is given as most given answer for this question. Let us see if the number D is correct or there are any other correct answer. So the correct answer will be number B, hydrophilicity. Hydrophilicity is not a QSAR parameter. What is QSAR? So QSAR or quantitative structure activity relationship is a mathematical relationship which correlates measurable or calculable molecular properties to some specific biological activity in terms of an equation. QSAR attempts to find consistent relationship between biological activity and molecular properties so that these rules can be used to evaluate the activity of new compounds. Understanding QSAR regarding electronic effects, steric effects and lipophilicity. Okay. So, physical or chemical parameters which are used for QSAR studies are hydrophobicity, means partition in terms. So, the hydrophobicity is the first parameter which is used for QSAR studies. And what are the terms or what are the parameters of hydrophobicity? How it is calculated? By the means of partition coefficient, pi substituent coefficient constant. Okay. There are steric parameters like Tuft constant, okay, and Varloop steric parameter. So, there are steric parameters for the QSAR studies, and we have electronic parameter like Hammett's constant and dipole moment. So, you do not have this QSAR in detail in your course, that much detail, and so I have just restricted it. There are various mathematical problems also related to this Hammett constant and dipole moment. I will try to give you one or two in the last class of next Tuesday. So, basically QSAR is like SAR. What is a SAR? Like we have a particular pharmacophore, we change the particular groups of the particular around the pharmacophore, and we see how the qualitatively that changes the compound's activity. So, QSAR is the same thing, but here we have a particular constants, we have several parameters through which we try to find a mathematical relationship of the calculable molecular properties. So, that is a QSAR. So, hydrophobicity, steric parameter, electronic parameters are the various physicochemical parameters for QSAR, but not the hydrophilicity. Okay. So, now I will move toward the next problem. This was your problem number 7. So, problem number 8, it says that what is the general term used to indicate the growth of new blood vessels? Your options are number A, biosynthesis, number B, apoptosis, number C, angiogenesis, and number D, metastasis. What is the general term used to indicate the growth of new blood vessels? Biosynthesis, apoptosis, angiogenesis and metastasis. Let me know in the chat box which of the following you think is the correct answer. What is the general term used to indicate the growth of new blood vessels? Biosynthesis, apoptosis, angiogenesis and metastasis. Biosynthesis, apoptosis, angiogenesis or metastasis. Which is the following term, general term, which is used to indicate growth of new blood vessels? Let me check the chat box now. So, number B, number C, number A. So, I have 3 different answers for this particular question. Okay, it's good. So, the correct answer will be number C, angiogenesis. So, biosynthesis is not a particular general term, it is a quite a big term. So, all the reactions which happens in our body, which is biosynthesis, terms can be terms as biosynthesis, one kind of. Apoptosis means the cell death. Metastasis is also related to that particular cell actions, and angiogenesis is the term, the general term which is used to indicate the growth of new blood vessels. So, molecular mechanism of angiogenesis is defined as the formation of new capillaries from pre-existing vasculature by migration and proliferation of endothelial cells. Okay. So, these are the typical example particular graphics for the particular angiogenesis. So, these greenish ones are the tumor cells. Okay, this one are the endothelial cell. Okay, and this bluish ones are the supporting cells. Red one are the platelets. This red dots, these red ones are the platelets. Okay, and this moon shaped one are the soluble proteases, and these are the basement membranes and extracellular matrix. Okay, these are the integrins. This red dots are the PDGF, and cross ones are the PDGF receptors, and the white vesicles are the VEGF, and bluish one are the VEGF receptors. So, you do not need to memorize these things, just remember angiogenesis term. This is indicates the growth of the new blood vessels. Okay. So, moving forward to the next question. Problem number 9 says the mitosis checkpoint is present in dash phase of cell cycle. Your options are number A M, number B S, number C G1 and number D G2. So, mitosis checkpoint is present in which phase of cell cycle? I have shown you the cell cycle in the very first question of today's session, where we were discussing about palbociclib or CD4K or 6K inhibitors. So, in the cell cycle where there are various checkpoints present. Okay. So, you have to identify. So, I will repeat the problem number 9 again. So, problem number 9 says the mitosis checkpoint is present in dash phase of cell cycle. Your options are number A M, number B S, number C G1, number D G2. In which phase of cell cycle does the mitosis checkpoint is present? the mitosis checkpoint is present in which phase of cell cycle? Let me check the chat box. So I have few answers. It says, so where should I start? So I think I have one answer only. It says S phase. Others also try. I will give you one or two more minutes. Just think about it carefully and let me know in the chat box in which phase of cell cycle mitosis checkpoint is present. Number A M, number B S, number C G1 and number D G2. So, others, did you answer the question? Yeah, I have few more answers. It says number M, number C, number A, number C. Okay, so I have different answer for this question again. So, let us check which of the following is the correct answer. So, the correct answer will be number A, M. So, in the M phase of cell cycle, mitosis checkpoint is present. So, now let us understand the cell cycle. So, this is the cell cycle. Through this cell cycle, the following diagram describes the different checkpoints of the cell cycles are responsible. So, cell cycle is basically the cycle through which a particular cell has to go through before it is divided into the two cells. Okay, so basically cell division goes before cell division a cell must go to this particular cell cycle. So, the cycle starts from this G1 or growth phase. Okay, so this growth phase, and first this is the G1 phase, and after the G1, in the G1 phase we have a G0 resting phase, which is a sub phase. Some cells can go to the G0 phase during the G1, and after when it is ready. And why the cells go to the G0 resting phase? See, the G1 is the growth phase. So, if the cell is not grown enough to go to the DNA synthesis phase or S phase, it will go to the G0 phase to rest, to take up all the nutrients and to be prepared for the ready to divide, and then it will go to the S phase. So, here at the end of the G1 phase we have a cell growth checkpoint. Okay, and this is the first checkpoint. What happens during this checkpoint? It occurs toward the end of the growth phase 1, G1, checks whether the cell is big enough and has made the proper proteins for synthesis phase. If not, the cell goes through the resting phase G0 until it is ready to divide. Then we have a S phase, which is a DNA synthesis phase. Okay, and during this synthesis phase we have in between a DNA synthesis checkpoint. So, what happens here? It checks whether the DNA has been replicated correctly. If so, the cell continues to the mitosis. Then we have a G2 phase, and then we have a M phase or mitosis phase. So, in the beginning of M phase we have this mitosis checkpoint. Okay, and it occurs during the beginning of M phase and checks whether the mitosis is complete. If so, the cell divides and the cell cycle repeats. Cancer can result when one or more these checkpoints malfunction. Okay, so this is the whole cell cycle, and we have this several checkpoints, and if a cell cancer results when one or more of these checkpoints malfunction. Okay, so this was your problem number 9. Now I will go to the next problem. So, problem number 10 says anti-ulcer drug omeprazole is number A, reversible proton pump inhibitor, number B, antagonist of H plus K plus ATPase, number C, agonist of H plus K plus ATPase, and number D, irreversible proton pump inhibitor. Let me check the chat box. Okay, so okay, let us discuss it because we have to complete another 10 set of question. So the correct answer of problem number 10 will be anti-ulcer drug omeprazole is number D, irreversible proton pump inhibitor. This is the structure of omeprazole. Okay. So omeprazole, or it is known as prilosec, and lansoprazole, which known as Prevacid, inhibit the proton pump effectively irreversibly, requiring synthesis of new enzyme protein. Omeprazole and lansoprazole are approved of the treatment for duodenal ulcer, may be used on conjugation with triple therapy, erosive esophagitis, gastric acid hypersecretory states including Zollinger-Ellison syndrome. So this is the proton pump inhibitor, the mechanism of action. Okay, so omeprazole, remember, it is a irreversible proton pump inhibitor, and as you can see, this has a benzimidazole pharmacophore. Okay, this is a benzimidazole pharmacophore. Also we have a pyridine ring. So as I have said, that you have to remember what are the heterocyclics are present in the particular, you have to you have to identify if the structure is given, what is the particular heterocyclics are present. I have discussed it in the second or third question. Today it is it. So this is the benzimidazole ring, this is the pyridine ring. So remember, omeprazole and lansoprazole are very important and very well known anti-ulcer drugs which are proton pump inhibitors. Okay, and they inhibit the proton pump irreversibly. So now moving toward the next problem. Problem number 11 says, which metal ion is present in the active site of cytochrome P450? Your options are number A, manganese, number B, nickel, number C, zinc, and number D, iron. So which of the following ions, metal ions, are present in the active site of cytochrome P450? All of you try to answer this question. Even if your answer is wrong, do not worry, but try to answer the questions. Which metal ion is present in the active site of cytochrome P450? Number A, manganese, number B, nickel, number C, zinc, and number D, iron. Which metal ion is present in the active site of cytochrome P450? It is a very well known enzyme, right? Cytochrome P450. We all have read it in our biochemistry. So now identify. Okay, let me check the chat box if somebody have answered it or not. Okay, so I have many answers actually. So yeah, it is like number D, number A, so number A and number D, in which number D have gotten the majority. So now let us discuss. So yeah, the correct answer will be number D. Iron is the metal ion which is present in the active site of cytochrome P450. So as you can see, this is the active site structure of the cytochrome P450. We have a porphyrin ring. In the center we have a iron. Okay, so the iron, first we have this iron 3 present, okay, and then after oxidation there is oxygen ion comes, and this oxygen, so what is the role of the cytochrome P450? Is basically to insert a oxygen atom in a particular bond. So, as you can see, there is a thiol bond and oxygen is inserting in between this S and hydrogen, sulfur and hydrogen, okay, and the cytochrome P450 catalyzes this kind of reaction, this oxygen insertion. And from where does this oxygen atom comes? From the atmospheric oxygen, okay, the O2. So, NADPH is the enzyme, and as you can see, this there will be this oxygen binds with this iron 3, and there is a electron flow, this kind of push pull mechanisms happens here, and iron 3 will convert it to iron 4 radical cation, which is a intermediate here, and after that we will have this kind of formation of this sulfur, this kind of oxygen O bond, and that is how the oxygen insertion happens. So, you do not need to remember the detailed mechanism, just remember what this cytochrome P450 does, basically insertion of oxygen between a bond, and what is the active site, there is a porphyrin ring here, and we have a iron in center of it, okay, and the oxygen comes from the oxygen, atmospheric oxygen O2. Okay, so, this was the problem number 11. So, now I will move toward the next problem. So, problem number 12 says which one of the following activities occurs following the stimulation of H2 receptors. Your options are number A, vasodilation, number B, uterine contraction, number C, bronchial smooth muscle contraction, and number D, enhanced secretion of hydrochloric acid in the stomach. Which one of the following activities occurs following the stimulation of H2 receptors? Number A, vasodilation, number B, uterine contraction, number C, bronchial smooth muscle contraction, number D, enhanced secretion of hydrochloric acid in the stomach. H2 receptor stimulation catalyzes which activity? Vasodilation, uterine contraction, bronchial smooth muscle contraction, or enhanced secretion of hydrochloric acid in stomach. Try to identify A, B, C or D, which of the following is the correct one. Even if your answer is wrong, it does not matter, but all of you please try to answer the question. Whatever you think, please write in the chat box. Even if your answer is wrong, it does not matter, but answering is very important. Let me check the chat box now. Okay, let us discuss. So, the correct answer will be okay, I hope my voice is clear, right? My voice is audible to you, and the PPT is visible. Okay, I am just checking to see if my internet connection is all right. Just can anybody confirm if my voice is audible to you, and also if you can see the PPT. Okay, thank you so much. Okay, now let us see. So, I had one answer, right? Number D, I think somebody have somebody have written in the chat box like number D will be the correct option. So, let us see. So, the correct answer for problem number 12 is number D. Very good. Enhanced secretion of hydrochloric acid in the stomach. H2 receptors are one type of histamine receptors. So, histamine receptors are of 4 types, H1, H2, H3 and the most recently H4. These are G protein coupled receptor. Activation of H1 cause contraction of smooth muscles in gut, uterus and bronchi. Contraction of bronchi leads to asthma. Stimulation of H1 receptors or smooth muscles in fine blood vessels cause muscle relaxation and the resulting the vasodilation may result to severe fall in the blood pressure. In CNS, H1 H2 receptors predominantly localized in the postsynaptic membranes. H3 receptors appear to function predominantly in the presynaptic receptor, probably histamine autoreceptors also be detected in some peripheral, and H4 receptors exhibit very restricted locations in intestinal tissues, spleen and immune systems like T cells, suggesting therapeutic potential of H4 blocker in allergic and inflammatory disease. So, here H2 receptors is not given that much of importance, but H2 receptors are very important, okay, because these are involved in the gastric secretion of hydrochloric acid in stomach. So, in between these four histamine receptors, H1 and H2 receptors are the major ones, okay. So, H1 receptors, so there is a comparison between two receptors. So, H1 receptors are histamine receptors that couple with GQ11 stimulating phospholipase C, histamine receptors H2 receptors which activate the adenine cyclase. So, the location is the periphery. The H1 receptors have the periphery in the body, adrenaline, medulla, vascular endothelium, heart and CNS, and the H2 receptors, they have in the location of H2 receptors are in the central nervous system and the peritoneal cells located in the stomach. Okay, antagonists are the first for H1 receptors are the diphenylhydramine, loratadine, cetirizine. These are used in the treating in the CNS diseases, and ranitidine, cimetidine, formatidine, netizidine. So, I have if you remember when I was discussing the PUD, the drugs which are used to treat the peptic ulcer disorders or diseases, I have mentioned the first class was like antihistaminics. Okay, H2 antihistaminics was the first class of, and these are gastric acid the stimulation inhibitor. They inhibit the oversecretion of the acids in the stomach. And the function of H1 receptor is the ileum contraction, modulates circadian cycle, itching and vasodilation, and they speed up the synaptic stimulation of the gastric acid secretion, smooth muscle relaxation, and all other, there are other also roles for this H2 receptors like inhibiting the antibody synthesis, T cell proliferation and cytokine production. So, remember the major activity occurs during the stimulation of H2 receptor is the enhanced secretion of the hydrochloric acid in stomach, and the inhibitors of this H2 histamine receptors are known as antihistaminics, are like for example ranitidine, cimetidine are very famous class of and drugs which are used to treat the PUD or peptic ulcer disease. So, now I will move toward the next problem. Problem number 13 says imatinib is an important anti-cancer drug which targets a protein kinase. What role does this piperazine ring has? This is the piperazine, this is the structure of the imatinib. Okay, we have this, there is a pyrimidine ring here in brown, we have a methyl group here present in the reddish one, and we have a purple spacer here, and the bluish one is the piperazine ring. So, what does this role of this blue piperazine ring here? Number A, it increases the water solubility. Number B, it increases the target selectivity. Number C, it increases target binding. Number D, all of the above. So, you have to identify what is the role does this piperazine ring has. Okay. Write in the chat box which of the following you think is the correct role of the piperazine ring in imatinib. Imatinib is an important anti-cancer drug which targets a protein kinase. What role does the piperazine ring has? Number A, it increases water solubility. Number B, it increases target selectivity. Number C, it increases the target binding. Number D, all of the above. Let me check the chat box. So I have one answer which says number D. Okay, let me check if it is the correct one and then let us discuss. So the correct answer will be very good, number D, all of the above. Increasing water solubility, increasing target selectivity and increasing target binding, all this three role is done by this piperazine ring. So what is the role of this imatinib? Imatinib is a kinase inhibitor. So kinase inhibitors are a very important class of the anti-cancer drug which I have discussed today in the beginning. Okay, I will discuss regarding this kinase inhibitors, how they work in more detail in the next summary class, so you don't have to worry. So this is a particular inhibitor of the BCR-ABL kinase. So as you can see, this is the BCR-ABL kinase in the greenish one. In this there is a substrate bind. Okay, and then we have this ATP which binds to this particular BCR-ABL. So when it is binding with this enzyme, this ATP is converted to ADP, and one phosphate of this gamma phosphate of this ATP is getting with the attaching with the substrate. Okay, and this particular transfer of phosphate group is catalyzed by this BCR-ABL kinase. So what does it happen? This substrate then after phosphorylation, this substrate will trigger the effector molecule and which will start a cell signaling, and this kind of signaling are responsible for many cancers. Okay. So imatinib or imatinib mesylate is a drug which will bind to the ATP binding pocket of this BCR-ABL. So now ATP cannot bind. If ATP cannot bind, the phosphate transfer cannot happen, and if this phosphate transfer cannot happen, the signaling will be totally closed. So that's how this particular kinase or any kinase inhibitor works. Okay. So there are, this is the kinase in this is the imatinib, and this various amino acid residues of the kinase enzyme which are interacting with this particular imatinib molecule, like this amino acids which are mentioned here, like histidine 361, isoleucine 360, the ASP 381. So all these amino acids are of this enzyme, where this is the active site binding of this particular inhibitor with this particular enzyme. Okay. So this particular enzyme, so there are various pocket of this BCR-ABL. So this particular pocket goes to the allosteric pocket, this goes to hydrophobic, and this goes to the adenine pocket. So this is the protein structure. This ribbons are the protein, and this is the in the purple, this is the molecule. All this thing I will discuss in the next class. So today I will skip this part. So remember, this is a particular imatinib is a particular anti-cancer drug where this piperazine ring increases the water solubility, target selectivity, and target binding. Okay. So now I will move toward the next question. Problem number 14 says cyclophosphoramide is commonly used in anti-cancer therapy. Which of the following statement is false regarding its action? This is the structure of the cyclophosphoramide, and you have to identify the incorrect or the false statement regarding its action. Number A, it is itself is a bisalkylating agent. Number B, it acts as a prodrug. Number C, it needs oxidative activation to show bisalkylation activity. Number D, the structure is metabolized to release acrolein. Which of the following statement is false regarding the action of cyclophosphoramide? Cyclophosphamide actually. Yeah. Identify A, B, C or D. Which of the following is the false statement? Number A says it is by itself a bisalkylating agent. Number B, it acts as a prodrug. Number C, it needs oxidative activation to show bisalkylation activity. Number D, the structure is metabolized to release acrolein. Now let me check the chat box. So, okay, I have few answers. Number D is given the answer. Okay, let me check if number D is correct one. So, the false statement regarding the cyclophosphamide is number A, it itself is a bisalkylating agent. Actually, it is not itself a bisalkylating agent. So, cyclophosphamide is an alkylating agent, but it widely used as DNA cross-linking and cytotoxic chemotherapeutic agent. It given orally as well as intravenously with efficacy. It is inactive in parent form. It must be activated to cytotoxic form by the liver cytochrome 450 liver microsomal system to 4-hydroxycyclamide or aldophosphoramide. Okay, the 4-hydroxycyclamide and aldophosphoramide are delivered to the delivering normal and tumor cell. Okay, so the cross-linking DNA results in the DNA synthesis. So, the mechanism of action, the main action effect of cyclophosphamide is due to the metabolite phosphoramide mustard. So, it is not an active form and it is not itself a bisalkylating agent. First, it acts as a prodrug. First, it has to be activated to the active form of drug, which is a bisalkylating agent. Okay, so I think now you know what is the, so it needs activation to show the bisalkylating activity. Okay, and phosphoramide mustard forms DNA crosslinks between the interstrand crosslinkings and the within the interstrand crosslinkings DNA strands at guanine 7 position. This eventually leads to the cell death. So, as you can see, this is a cyclophosphamide off target oxidation. Okay, this is converted to this active form 4H cyclophosphamide, okay, by the cytochrome P450 enzyme, and then it converts to the aldophosphamide and it goes metabolized to acrolein, and then it has the phosphoramide mustard. This phosphoramide mustard is the active form, which is the bisalkylating agent, which is the reason for the DNA and protein alkylation and cell death, and this acrolein give rise to the tissue toxicity. Okay. So, number A was the false answer regarding the cyclophosphamide. So, now problem number 15 says, for the better bioavailability of a drug, what should be the maximum number of rotational bonds? Number A 10, number B 7, number C is 5 and number D 13. So, for the better bioavailability of a drug, what should be the maximum number of rotational bonds? A, B, C or D? Try to answer in the chat box. For better bioavailability of the drug, what should be the maximum number of rotational bonds? Try to answer this question in the chat box. Number A 10, number B 7, number C 5 and number D 13. Let me check the chat box if there are any answers. Okay, I have few answers. Okay, bisalkylating agents is the agent which will give rise to alkylation twice. So, let us say there is a particular compound and there is a bisalkylating agent will add the alkylate it twice. Okay, I will give you the detail in the next class about the bisalkylating agent. If you have any doubt after that class, then you can let me know. Okay. So, basically it will give rise to the alkylation of a particular starting material twice. I will give you the detailed mechanism in the next class, and then you will have the particular vision about this bisalkylating agents. Okay, so for this question I think I have various questions. Number A, number C, very many answers are given. So, let us check. So, the correct answer is number B, 7. 7 should be the maximum number of rotational bonds for better bioavailability. Okay. So, a rotable bond is defined as any single non-ring bond attached to a non-terminal non-hydrogen atom. Amide CN bonds are not counted because they have high barrier of rotation. So, as you can see, there is an example. So, these two drugs are for the antihypertensive drugs. This is the atenolol, this is the propanolol. If you count the number of biorotable bond, so you will see that, so for atenolol we have 8 number of rotational bond. These red ones are the rotational bond. This 1, 2, 3, 4, 5, 6, 7, 8. So, these are the rotational bonds of atenolol which have 8, and propanolol we have 6, 1, 2, 3, 4, 5 and 6. Okay. So, if you can see the bioavailability for propanolol, it is 90%, which is much better than the 50% of the atenolol. So, the maximum number should be 7. If the number of rotational bond exceeds 7, bioavailability will decrease, and if it is lower than 7 or equal to 7, the bioavailability will much better. Okay. And it should be a non-terminal non-hydrogen atom bond. So, a bond connected to only hydrogen is not counted, and it should not be terminal. Okay. And amide CN bond, so this is the amide CN bond, this one is not counted because it has a very high source of high barrier of rotation. Okay. So, now I will move toward the next problem. Number 16 says method of action of aromatase inhibitor is number A, irreversible inhibition, number B, reversible inhibition, number C, suicide inhibition, and number D, uncompetitive inhibition. So, what is the method of inhibition, method of action of aromatase inhibitor? Is it irreversible inhibition? Is it reversible inhibition? Is it suicide inhibition, or is it uncompetitive inhibition? What is the method of action of aromatase inhibitor? Is it irreversible inhibition? Is it reversible inhibition? Is it suicide inhibition or uncompetitive inhibition? Let me check the chat box. So, I have answers number B, number D. Okay, let me check which one is correct. So, the correct answer is number C, suicide inhibition. Okay. So, aromatase inhibitors are suicide inhibitor. So, this is the So, how the aromatase inhibitor works. So, androstenone, dione testosterone, okay, these are converted to estrone and estradiol by the means of the aromatase. Okay, and this estrone and estradiol, estrone is related to with the ER activation and estradiol with the generation of E234 quinone. Okay, and this ER activation give rise to proliferation of mutated cell, and generation of E234 quinone is related with the DNA alkylation and depurination, and both of these give rise to the cancer. So, aromatase inhibitor inhibit this aromatase enzyme, and that is how this is a very important class of anti-cancer drugs. So, this is the particular mechanism of action or active site mechanism for the aromatase inhibitor. You do not need to memorize this, I just given for your knowledge. So, we have this this is the active site. Okay, we have a iron in the active site of aromatase inhibitor. First, in presence of oxygen, when it is the iron 2. When oxygen binds with the iron, it is oxidized to iron 3, and we have this oxygen radical, and then we have this electron transfer, single electron transfer, and we have this O minus. This O minus actually attacks on this carbonyl carbon of this starting material and or the substrate, and then we have this kind of active site mechanism. Okay, and that is how this particular testosterone and all this converted to estrone and estradiol. So, you do not need to memorize, just if you remember the active site mechanism, just remember it has the iron in the active site, iron is in the iron 2 state, okay, and which is converted to iron 3 by the means of the oxygen binding. So, aromatase inhibitor, remember it is a suicide inhibition is the method of the action of aromatase inhibitor. So, now I will go to the next problem. Problem number 17 says statins work as number A, HMG-CoA synthetase inhibitor, number B, HMG-CoA reductase inhibitor, number C, squalene synthetase inhibitor, and number D, acetoacetyl-CoA thiolase inhibitor. So, statins work as which kind of inhibitor? Number A, HMG-CoA, number B, HMG-CoA synthetase inhibitor, number B, HMG-CoA reductase inhibitor, number C, squalene synthetase CoA inhibitor, and number D, acetoacetyl-CoA thiolase inhibitor. As you can understand, this all is related with your cholesterol biosynthesis. I have mentioned cholesterol biosynthesis pathway, all these enzymes which are mentioned here, HMG-CoA synthase, HMG-CoA reductase, squalene synthase, acetoacetyl-CoA thiolase, all these inhibitors are, all these enzymes are basically involved in the cholesterol biosynthesis. So, inhibitors of this enzyme will work as a anti-cholesterol drug, the drugs which treat the cholesterol problems. Okay, statins in one of those. So, you have to identify statins work as which kind of inhibitor. Statins works as which kind of inhibitor? Which enzyme does statins inhibit? Let me check the chat box. Yes, so I have few answers. Number B. So, number B is the only answer. Okay, I have number C, one number C also. Let me check which is the correct answer. So, the correct answer will be number B, HMG-CoA reductase inhibitor. So, now HMG-CoA reductase inhibitors or statins, the mechanism of action is analog of HMG-CoA, competitive inhibitor of HMG-CoA reductase, the rate of limiting step in the cholesterol synthesis, reduction reduced hepatic cholesterol levels in low density lipoprotein receptor expression, which increases the removal of the LDL in the blood. Reduction of hepatic cholesterol result in the decrease in the VLDL synthesis and secretion. Adverse effect are hepatotoxicity less than 1% of patient, myopathy 2 to 10% of patients, rarely lead to rhabdomyolysis and contractions in pregnancy. So, this is the HMG-CoA reductase inhibitors or statins are a very important class of a drug which are used for the treatment for the cholesterols. Okay. So, now I will move toward the next problem. So, problem number 18 says drug resistant of trimethoprim is generated due to number A, efflux pump, number B, sequential blocking, number C, mutation in target, and number D, insufficient drug concentration. So, trimethoprim is a very important drug. Okay. So, the drug resistance of trimethoprim is generated due to efflux pump, sequential blocking, mutation of target, and insufficient drug concentration. Trimethoprim is a very important class of antibiotics. I have discussed it in your last to last class, I think, where I was discussing the antibiotic drugs. So, you have to identify the drug resistant of trimethoprim is generated due to which mechanism: efflux pump, sequential blocking, mutation in target, or insufficient drug concentration. Drug resistant of trimethoprim is generated due to which mechanism? Let me check the chat box now. Okay. Number D. Number D is most given answer. Let me check, then let us discuss. So, the correct answer will be number C, mutation in target. So, let us say you have a protein. So, the mutation can occur here and this results in the conformational change which changes the binding pocket of enzymes. So, this is how this is not a very good binding anymore. For example, resistant to antibiotic trimethoprim derives from a single mutation of a single amino acid of target enzyme, which is dehydrofolate reductase. So, these properties of singly mutated enzyme differ from those of normal enzyme, but it still binds to the substrate dehydrofolate. So, this is the pteridine and para-aminobenzoic acid which give rise to dihydropteroic acid. Then dihydropteroic acid goes to the dihydrofolic acid and tetrahydrofolic acid. So, it is the your folic acid metabolism or folic acid synthesis of the bacteria. There are various enzymes which are involved like dihydropteroid synthesis, dihydrofolate synthesis, dihydrofolate reductase. Trimethoprim, it inhibits the dihydrofolate reductase, that is how it shows its antibacterial effect. But there is a single point mutation of the particular enzyme, the dihydrofolate reductase, which give rise to the resistance with trimethoprim. The single point mutation happens like this enzyme will bind with the substrate, but it will not, the trimethoprim cannot affect this particular enzyme. So, that is how the particular drug resistance of trimethoprim is generated. Okay. So, now I will move toward the next problem. Problem number 19 says aminoglycoside resistance is due to number A, target mimicry, number B, target overproduction, number C, efflux pump, and number D, lack of the electrical potential gradient across the bacterial membrane. Aminoglycosides are also very important class, one of important class of the antibacterial drugs. So, you have to identify which is the mechanism of resistance for this aminoglycosides. Number A, target mimicry, number B, target overproduction, number C, efflux pump, and number D, lack of an electrical potential gradient across the bacterial membrane. Which is the mechanism of resistance of aminoglycosides? Tell me in the chat box which of the following you think is the correct answer. Target mimicry, target overproduction, efflux pump, or lack of electric potential gradient across the bacterial membrane. Now let me check the chat box. So various answers, number B, number D, number B. So number B and number D are the most given answers. Let me check which of the following is the correct one then. So the correct answer will be number D, lack of an electrical potential gradient across the bacterial membrane. So aminoglycoside or aminoglycosides is a medicinal or bacteriologic category of traditional gram-negative antibacterial therapeutic agents that inhibit the protein synthesis and contain as a portion of the molecule an amino modified glycosides. Okay, aminoglycosides antibiotic display bactericidal activity against gram-negative aerobes and some anaerobic bacilli where resistance has not yet arisen, but generally not against gram-positive anaerobic gram-negative bacteria. Aminoglycoside antibiotic resistance can arise from lack of quinones that mediate the drug transport from a lack of an electrical potential gradient required to drive the drug across the bacterial membrane. So one of the ways which can occur by changing the net charge on the membrane. So if you can example here is the phosphoglycerol, the membrane in the changed in the lysyl phosphoglycerol groups, then it converts to an anionic group to a cationic group. So phosphatidyl glycerol, if it is converted to lysyl phosphatidyl glycerol, then it is converting the anionic group to cationic group, so changing the permeability of a drug. So if the particular gradient of a cell membrane changes from anionic to cationic, the drug which were first entering to the particular membrane, now it will not get entered because it will repel. This way drug have some charge can actually now repel. So aminoglycoside antibiotic, for example, because ineffective because quinone that mediate the drug transport, or from the lack of quinone that mediate the drug transport, or from the lack of an electrical potential gradient required to drive the drug across the membrane. So these two can result in decreased uptake of aminoglycosides, which is going to result in the resistance. Okay. So now the last question of today's session, it says, which of the following physicochemical properties is least commonly considered in QSAR studies? Number A, hydrophobicity. Number B, electronic influence of substituent. Number C, dipole moment. Number D, size of the substituent. So which of the following physicochemical properties least commonly considered in QSAR studies? Tell me in the chat box which of the following you think is the correct answer. Which of the following physicochemical properties is least commonly considered in QSAR studies? Hydrophobicity, electronic influence of substituent, dipole moment, and size of the substituent. Which of the following you think is the correct answer? Let me know in the chat box. Take 1 minute. This is the last question of today's session, so I want all of you to try to answer this. Hydrophobicity, electronic influence of substituent, dipole moment, and site of the substituent. Which of the following is least commonly considered in QSAR studies? me check the chat box if there is any answer. Okay, so I have few answers, many answers I think. Number D, number C, number A, number D. Okay, so I have various answers. Let me check which is the correct answer. So the correct answer will be number C, dipole moment. So dipole moment is least commonly considered QSAR studies. So the rationally for QSAR studies is a quantitative structure activity, or QSAR correlates measurable or calculable physical and molecular properties to some specific biological activity in terms of an equation. Once a valid QSAR has been determined, it should be possible to predict the biological activity of the related drug candidates before they are put through the extensive and time consuming biological testing. In some cases, only computed values need to be known to make an assessment. So QSAR, there are various parameters like geometrical, thermodynamic, electronic, constitutional, topological. So what happens basically in short in the QSAR, okay, so QSAR there is a calculation happens. So let us say there are 100 drug candidates and we have a particular lead drug candidate which we need to we have some specific QSAR values, okay, the parameter values. So now all the drug candidates are calculated, those parameters value, and the values are closer to the lead compounds value, will be only tested for the biological screening. So now if we want to test all the 100 compounds for biological, so it will take a long time and it will be expensive. But if you have the QSAR parameters number values, so we can reduce the 100 to let us say 15 to 20 molecules which have a very much comparable QSAR values with the lead molecule. So now this 10 or 20 molecules only be tested for the biological study. Okay, so that is how it will be easier to assess the drugs and it will be less time consuming for to address a new drug. Okay. So this was the last question of today's session. So today what we discussed a class of anti-cancer drugs mostly. Then we discussed about your proton pump inhibitor kind of drugs, mean anti-ulcer drugs. We discussed about cholesterol biosynthesis and drugs related to that, and we discussed few question about the antibacterial biosynthesis. So I will share my email ID in the chat box, okay, which I didn't see. 0. So this is, yeah, I will share the slide. Okay, let me just finish this. So if you go to the SWAM portal, so you will say where there is a assignment section in the lower in that portion, you will have a problem solving session Excel sheet. In that Excel sheet, all the links for the previous classes and today's classes I will upload it soon, and also the slides are present in that Excel sheet. If you cannot find the Excel sheet on the SWAM portal, please let me know. In the next class I will show you where to find it. Okay. So it is in the in the lower portion of the assignment section. You will find all the slides of our previous classes and all the videos. So today we finished our week 12 course content. So all the courses, all the weeks we have done. So now I will take 2 more classes. One will be on this Sunday 15th October from 11 and it will be 11 AM. So it will be 1 hour class only. In that 15 October's 1 hour class, I will summarize the whole course. No question will be given on in that class, only I will summarize the course. I will introduce you few more topics related to the biology and drug development, which will help you to understand the course much better. So please join that class, and the last class of this session we will have on the next Tuesday 17th October, where I will give you few questions, one question from each week. So we will solve 12 questions. I want to see if you have prepared all your weeks very clear, if you are prepared with for your annual examination. So I will give you 12 questions for each one question each week, and I will discuss few more topics. So it will be 1 hour of question answer session, 1 hour of your understanding summary session for the next Tuesday. So thank you all for joining today's session. I will close the session for today. So next class we will be on Sunday or from 11 AM. So please join that class. So I will close the session for today. If you have any doubt, any query, let me know in this particular email ID. Okay. So thank you all. I will close the session for today.